everyone, and a warm welcome back to our Evolution and Ecology seminar series. My name is Elizabeth Duxbury, and I'm a postdoc in the research group of Alexei Maklikov at the University of East Anglia, and I'll be hosting today's session. As always, we will have a Q&A session directly after today's talk, so please post your questions in the dedicated Slack channel. And with that, it is my great pleasure to introduce today's speaker, Professor Pat Monaghan, the Regius Chair of Zoology in the Institute of Biodiversity, Animal Health and Comparative Medicine at the University of Glasgow here in the UK. Pat is an evolutionary and behavioural ecologist whose research explores how early life conditions such as stress exposure can shape the life history trade-offs between growth, reproduction and senescence. And she is actively involved in understanding the underlying mechanisms involved, with a particular interest in telomere dynamics. During an internationally recognised career, Pat's research provided the first experimental demonstration that the maternal condition of birds can affect the sex ratio of their offspring, and she has also extensively studied both wild and captive populations in elegant longitudinal studies. This work has revealed the complex effects of environmental change, parental age, and early life nutrition on longevity, both in zebra finches and also in a variety of other taxa. Pat's research is continuing to make important contributions to our understanding of life history evolution and the importance of early life experiences on later life. And she is the recipient of several highly prestigious distinctions, including Fellowship of the Royal Society of Edinburgh and the Medal from the Association for the Study of Animal Behaviour. Impressively, Pat is also the recipient of 2 European Research Council advanced grants, the second of which he was awarded just last week, to study the effects of maternal age on offspring performance. Today, Pat will present her talk entitled Stress, Exposure and Longevity, Organismal and Cellular Level Effects at Different Life History Stages. So Pat, thank you for accepting our invitation, and over to you.
Thank you very much, Elizabeth. I hope you can still hear me okay. Well, thank you for your very nice introduction, and thank you for inviting me to speak this evening. I'm going to talk about stress exposure and longevity, and I'm going to talk about the organismal level and to some extent also the cellular level effects. What I thought I would first do is just say a little bit about my recent research focus. I'm especially interested in environmental effects on individual life histories in a wide range of different contexts. One thing I have been particularly interested in are trade-offs between growth and longevity, and how the tempo of growth may influence longevity and health in later life. I've also been interested in particularly these long-term effects of early life conditions that can, for long-lived species, extend across decades. And that led me to get interested in these factors which are influencing variation in aging rates. So my work now combines functional and mechanistic approaches. As Elizabeth said, I'm a behavioral and evolutionary ecologist, so I'm especially interested in function and fitness effects and so on. But I've been increasingly interested in the mechanistic processes that might underlie the organismal level effects that we see. Why am I interested in mechanisms? I think it helps us identify trade-offs and not make spurious connections that may not actually be real trade-offs. And it also helps us to understand the constraints under which individuals might be operating, but also that influence the path of evolution. the work that I'm involved in is now, it's predominantly lab work, but I still am very interested in field work, and I've worked on a wide range of species together, of course, with students and postdocs, and I've been very fortunate to have worked with some extremely good students and postdocs. And the kind of work that we do is both observational work and experimental. And so I've had many collaborators on whose expertise I have relied. I've worked with a wide range of species in different contexts, but my work is predominantly on birds. I'm especially interested in birds because many of their life history attributes are particularly interesting if you're interested in long-term effects. They live quite long. If you're interested in reproductive strategies and mechanisms, working with an egg-laying species allows you to do experiments that are much less invasive than they would be, for example, in a mammal. I'm interested in many attributes about the exceptional longevity of birds in relation to their body size. But I am interested, of course, also in other taxa. What I'm going to do in this talk, the focus of this talk is I'm going to talk about these effects of stress exposure at different life stages on longevity. And I'm going to talk about some of the roots, of course, not all by any means, but some of the roots and mechanisms that underlie these effects, and particularly some physiological aspects. And I'll say a bit about telomere dynamics, as Elizabeth mentioned. Now, why telomeres? Well, of course, I'll say a bit more about telomeres shortly, but in the context of the organismal level effects of stress and telomere dynamics are linked, and they're in fairly recent years, there's been a lot of interest in the link between environmentally or internally generated stress and the dynamics of telomere change. The connection, as the paper here by Sapolsky mentions, was originally unexpected, but actually the more mechanistic processes that are uncovered, the more we understand how stress may impact telomere biology and thereby influence health and longevity. Now, much of the focus of the link between stress exposure and longevity has looked at long-term consequences, and primarily in a biomedical context. There's not been a huge amount of attention in the context of evolutionary or behavioral ecology. But those of us who are interested, who come from a more biological, especially zoological background, would expect that the outcome of individuals of stress exposure will vary with the life history stage, the life stage at which they experience the stressor. It may experience with the organism's life history, and also with the severity of the stressor. And I'm interested in these effects in particular. If you're experiencing stress very early in life, the phenotype may be permanently adjusted. If you're experiencing it in adult life, the dynamics may be different from the phenotypic changes that can occur in early life. And if you're a long-lived as opposed to a short-lived species, evolution will have varied the extent to which, if you like, you care about long-term effects, because your life history may be such that you don't experience them in terms of their fitness consequences. And also the severity of the stressor itself is going to be very important. So the relationship between stress exposure and fitness is not a simple one. And in fact, there's a general movement towards the idea that there may be an optimal level of stress exposure to ensure appropriate resilience given the particular set of environmental circumstances that the individual experiences in its life, and also to maximize the fitness benefits. So I'm going to come back to this, that no stress exposure may make individuals less resilient. And of course, I mentioned the severity of the stressor. Too much stress is unlikely to be a good thing either. So there may be optimal levels. And those optimal levels might vary amongst individuals and also amongst populations. And this idea hasn't been studied very much from an evolutionary ecology perspective, as I've said. But here, for example, is a paper by Robert Sapolsky on this idea of an optimal level of stress exposure. But that's a discussion from a neurobiological perspective rather than from an ecological one. But it's a very interesting paper. Now, I've had a long-term interest in early life adversity and its effects on fitness. And I've been involved for many years in a study of this species here, the red-billed chough. We have a population of this bird on the Inner Hebridean islands of Islay and Colonsay, and that's now the only population in Scotland. So it's a bird of considerable conservation concern in Scotland and in the United Kingdom in general. We've been looking at the Scottish population, which particularly the one on Islay, but also to a lesser extent, because it's a much smaller population, on Colonsay, in a long-term study since 1981, particularly with Jane Reid, who's now in Norway, Eric Bignell, who lives on Islay, and David McCracken, who's an agricultural biologist, and many others. So we've been tracking individual life histories in this species. I'm not going to talk about all of that. A lot of it's, of course, concerned with conservation biology, but I want to draw your attention to one of the things that got me interested in early life adversity. One thing that came out of the study of individual life histories that we've been doing in this species in a series of earlier papers on its ecology, early life conditions, now that can refer to being born in a poor year or being born in a poor area of the island, have a profound influence on subsequent performance. What we found is that poor conditions are associated, obviously conditions influence breeding performance. I'm now talking about post-fledging conditions. Being born in these poor conditions and surviving until you fledge is associated with reduced survival during the first year of life. So even having made it through that fledging, that nestling period, those individuals that survive the poor conditions, who are probably sometimes the best of those experiencing the poor conditions, they still have relatively poor survival in their first year of life compared to individuals that did not experience those conditions. Even after that relatively high first year mortality, they still have a reduced probability of recruitment to a breeding population. They don't recruit until they're 2 to 3 years old. Then, even having made it to recruitment, they have a reduced number of breeding years and a reduced lifetime reproductive success. And these effects, which were particularly interesting for me, seem to be irrespective of where the individuals settle on the island later in their life, and to some extent irrespective of later life conditions. So these early life conditions appeared to be a legacy for these individuals that cast a shadow over their subsequent performance. And so I was very interested in that effect in particular. Now, I've been involved in a number of studies, as Elizabeth mentioned, with the zebrafinch. It's a fantastic species for studying in the laboratory. And I'm going to show you a number of results of some of the work that we've been doing with this species. The 4 individuals that I've put up there have been the kind of dream team that I've worked on with these projects, but not the only people, but they've played a big role. Winnie Bonner and Valeria Marasco as postdocs, Kate Griffiths as a technician, and Britt Heidegger also worked with me as a postdoc on some of this work for part of the time. Because we have these birds in captivity, we're able to follow through individuals from when they're a chick to when they're an adolescent to when they're an adult. These are males here. And then in old age, when they show the phenotypic changes that are characteristic of old age. And I always feel that it's important when you're doing studies on captive species, very difficult to look at fitness, for example, in captivity. We can look at indices and correlates, but I also feel it's very important to have related experimental and correlative studies in the field on other species to help inform subsequent lab experiments, but also to ensure that the results are applicable in other contexts. Now, during development, individuals can be exposed to stress, and it can induce permanent changes to the phenotype. A persistent signal that the environment is challenging can come either directly to the individual from the environment, or it can come via effects of parental state. Now, in the context of stress biology, one of the things that happens is a key aspect of the physiology of vertebrates is the HPA axis, which controls the stress responses, and that can become more reactive. And we might think that may well be an adaptive response to a dangerous situation. So here's some work that I was involved in some time ago with another postdoc, Karen Spencer, who's now in St. Andrews, on the zebrafinch. And one of the things we were interested in was how early life exposure to elevated stress hormone levels influences the functioning of the HPA axis later in life. So what I'm showing you here on this graph, this is the standardized capture stress exposure response that is often done on birds to look at the acute stress response. So this is the corticosterone level in the bloodstream in relation to time since capture. And the dark line here are the control birds, and the dotted line is a group that we treated when they were nestlings, such as to elevate their corticosterone levels by a daily dose of corticosterone. This is the acute response, not measured during that treatment period. That only lasted until 30 days. This is the acute stress response when these birds were young adults. Now, what you can see in the control birds is the typical response. There's a rise in the baseline corticosterone level in the bloodstream to a peak level, and then it falls off. What you can see in these treated nestlings who are now young adults and haven't received any treatment since the end of the nestling period is that they're more reactive. They secrete a higher level, faster, of corticosterone into the bloodstream, and it tails off more slowly. So what that means is that, as far as we're aware, probably for much of their adult lives, these individuals are more responsive to stress. When they encounter a stressful event, they secrete more corticosterone. So they're always more exposed to corticosterone than the control birds would be. And they're therefore very reactive. Now that has consequences. This increased reactivity coming from the increased stress hormones in early life, we found has long-term consequences for lifespan. So here you see The control in black, the corticosterone treated individuals in red. Remember, they only received treatment for a very short period early in life. This is survival during the first year of life. There's no difference between the control and the corticosterone treated birds. But as you can see, as the individuals grow older, and particularly as they start to become relatively old, their mortality risk is substantially higher. In other words, they seem to be aging at a faster rate, and so their lifespan is reduced. Now, I'm going to come back to these birds briefly later, but so they were treated when they were nestlings, and then they were kept in a benign environment in their laboratory, and this is what happened to their longevity. Now, why did they adjust their phenotype? Why do individuals do that? Why aren't they as stress reactive as they can be? Well, as we've seen, there may be a lifespan penalty associated with exposure to elevated stress hormones during life. And if you're in a more dangerous environment and you're more at risk of mortality, being more stress reactive may be, in fitness terms, beneficial. You may be more resilient and you may be more able to escape predation, for example, because you respond very quickly. And that's traded off against these long-term costs. Now, I mentioned that stress and telomeres are a good reason to think that these are linked. So we were asking the question, also, does increased telomere loss contribute to this stress-induced reduction in lifespan? So I'll first briefly say a few quick words about telomeres. This telomeric system of chromosome protection is highly conserved across the eukaryotes. It's a sequence of DNA that occurs at the ends of the linear chromosomes, and it caps and protects and identifies the chromosome ends. Across all of the chordates, the sequence, the telomeric sequence is TTAGGG, and that's a repeated sequence in tandem repeats across the telomere, along with some proteins and other factors. Now, the important thing is that the pattern of loss and restoration of telomeres with age varies across species. It also varies among individuals and tissues within species, and the pattern of loss and restoration is influenced by the environmental conditions. Why did telomeres evolve? They solved two problems with linear chromosomes that the eukaryotes evolved, probably primarily for the advantages associated with sexual reproduction. The telomeres identify the chromosome ends and prevent any activation within the cell of DNA repair processes, so they maintain stability of the genome, preventing end-to-end joining of chromosomes, for example. The process of DNA replication across a linear chromosome is such that there's incomplete replication at the end of the lagging strand of DNA. That means every time a cell divides, some of the telomeric sequence, in this case, rather than the coding sequences, are lost from the chromosome end. So the telomere is getting shorter, but the individuals are not losing coding sequences. And once the telomere becomes critically short, it becomes dysfunctional. So telomeres are considered to be one of the hallmarks, telomere loss considered to be one of the hallmarks of aging. This is, of course, a series of self-portraits here done by Rembrandt, charting the inevitable phenotypic decline that we and other organisms see during aging. Telomere length determines the replicative potential of cells. If telomeres are allowed to decline, then the cells can only divide a finite number of times before they become really effectively senescent, and they cease to divide and they may die. So telomere loss controls cell replicative potential. Increased telomere dysfunction in the absence of restoration has serious consequences for health in later life. It's linked to a whole raft of age-related diseases, cancer, cardiovascular disease, and so on. And variation in length and loss rate is linked to longevity within many, but not all, taxa. And there's also been experimental studies recently showing that experimental increase in telomere length can slow aging and increase longevity. So one of the experiments, I'm switching here now to a little field project, which involved another postdoc, Catherine Herborn, which we did on a long-lived seabird, the European shag here. And we repeated that experiment that I showed you on the zebrafinch of dosing individual chicks in the nest with corticosterone. We looked at their acute stress response in the same way I showed you with the zebrafinches. It was also elevated in the same way as a consequence of the experience as a nestling. And we also looked at their telomere length at 30 days of age, while they're quite a well-grown nestling. They haven't quite fledged at that point. And here you can see the average telomere length in the controls and the average telomere length in the stress treated individuals, and it's substantially less. Now, I should say I'm going to show you a number of graphs that include telomere length on the vertical axis. It's measured in most of these graphs, in fact, all the ones I'm going to show you today by qPCR. So the measurement is a relative measure, a TS ratio, and they're measured in blood cells, red blood cells. Birds have the advantage that their red blood cells are nucleated, and so we can extract DNA from the red blood cells. So in shags then, the exposure to stress is shortening telomere length, and we know in the shag that telomere length at an early age is predictive in the long term of telomere length and also linked to mortality pattern. Now, I want to come back to this graph that I showed you earlier of these zebrafinches in this captive environment. And I showed you that they had a shorter lifespan in the captive environment. Now, the captive environment is benign. One thing we didn't do in this experiment was to expose these individuals to a more stressful environment when they were adults. It might have been that had we done that, we may have found a different pattern. We may have found actually that these corticosterone treated individuals in a stressful environment may have done as well as the controls, or even better than the individuals who had not had this early life stress experience. We had done experiments of that kind with a different kind of stressor, a temperature stressor, and found such an effect that a mismatch can actually wipe out, or in fact can actually cause the non-exposed individuals to do worse than the exposed individuals. So there are complicated things going on that relate to the conditions in early life and the conditions in adult life. So what happens in adult life is important. But so also, not all individuals experience stress in early life. So I'm going to tell you now about an experiment that we started in 2011, a long-term experiment. We started with young adults this time, and these birds were exposed to really, they lived in a challenging environment. They were repeatedly exposed to challenging conditions that elevated their corticosterone levels. Those conditions were unpredictability in food availability, and that increased baseline corticosterone levels for part of the time. They weren't on a caloric restriction because it was only a short part of the day that the food was not available, but it was unpredictable. The rest of the time they had ad libitum food. And what we've looked at a number of different things in this experiment, but I'm going to show you what effect this had on survival at different stages in the subsequent lives of these individuals. The first thing, though, before I go on to survival, it did affect their breeding performance. Here is the breeding performance of these individuals. They were all females in this experiment, and they were bred with control males. Here's the breeding performance when they were 6 months old, young. The stressed individuals did not do quite so well as the control individuals. But you can see this is later when they're really quite old for a zebra finch. You can see that the age-related decline in performance, breeding performance in this benign captive environment, or relatively benign in the control group, is very marked. In the stress-exposed group, though, the decline is less so. So there's some evidence of them being a bit more, aging a little bit more slowly in terms of reproductive performance. What about longevity? Well, again, the stress group is in red, the control group is in black here. This is survival during the first year of life. No difference, so that they're not, being really heavily impacted. This is a mildly stressful environment that we have them in. However, when they were middle-aged, somewhat to our surprise, I have to say, we found that actually survival was better in the individuals living under a mildly stressful environment. They seem to show an age-related increase in mortality that was much less marked during middle age than the control individuals did. And then when they were old, though, that survival advantage of these stress-exposed individuals was, well, the survival advantage disappeared. And actually their survival, if anything, was somewhat below that of the control birds in old age. So a different pattern depending on which stage we were looking at these individuals in. Now, of course, there are many things going on here, which I don't have time to go into, but what I want to show you now is we've also been looking to what happened to the telomere change in these individuals. And we can show you that on this graph here. Here's in young adulthood, the period when I showed no difference in survival. This is telomere loss during that period on this axis. This is the means for the control and the challenge group. And you can see that there was no difference in telomere loss. If we look at the mid-adulthood period, where the survival was better in the individuals in the challenged environment, you can see that actually their telomere loss on average was less than that of the control individuals. And then in this period, when the survival pattern changed and was possibly reversed, we actually found that these challenged individuals showed actually a slightly higher telomere loss than the controls. So what seemed to have happened to these challenged individuals, that they didn't escape senescence, they showed a somewhat delayed senescence, particularly in middle age, and their reproductive performance was also affected. But when they were old, things caught up with them again, if you like, at least in terms of their telomere dynamics. Now, one other thing that we also found was that when these females were young, though there was an intergenerational effect, those living in more stressful environments had sons with shorter telomere lengths at fledging. And again, in the zebra finch, we know that Telomere length at fledging is predictive of subsequent mortality. So here you can see in this graph the maternal environment, those living in the control conditions, in the more stressful environment. This is the telomere length of their sons and the daughters in blue, the sons in green, of their offspring at fledging. You can see in the control birds no difference, but in these females who had been living in the stressful environment, I should say that stressful conditions were not present while they were breeding. We put them on the same breeding regime as the controls. So it's not feeding of the offspring that's a problem. And what you can see is that the decline in telomere length in the sons is much more marked. This is when these individuals were young. This effect could be a pre or a postnatal effect. However, importantly, it disappeared when this difference between the control and stressful groups disappeared when these mothers were old. Even when we looked at the same individuals when they were young and when they were old, we got the same within individual effects. So what happened when they were old is there was a much greater effect of maternal age than the environment effect. And I'm going to show you that shortly. So the environment effect was overtaken and submerged by the effect of maternal age on telomere length. So that maternal age effect brings me on to the final kind of area I want to talk to you about, which is the effect of parental age on offspring longevity. Reduced offspring longevity in relation to parental age has been reported in many taxa from rotifers to humans. It doesn't always occur, but it's been termed the Lansing effect. And we've written a couple of reviews just recently of this effect. One with Alex Maklakoff, which is in Trends in Ecology and Evolution, which deals with a more obviously ecological and evolutionary perspective. And one in Proper-Oysalt, which dealt much more with the gametic effects and gametic aging and the role that might play. There are multiple potential causes of this. Some of them may be prenatal and gametic, and some of them postnatal, related to the environment, for example, that parents can provide for their offspring. And maternal and paternal effects have been reported, but they don't always occur. And also there's evidence that where they do occur, these effects may cross the generations. Now, I just want to show you a couple of examples. This is from our captive zebra population. This is offspring survival. Sorry, that should say this percentage here. That's a little bit missed off the legend there, but I just want you to look at the decline. These are offspring from young mothers in red here and offspring from older mothers. You can see the decline in survival with age is much steeper in the offspring from the older, the proportion surviving is much less in the offspring from the older mothers than it is in the young mothers, so higher mortality in the old mother offspring. And here's the same kind of figure for a mouse population. In this case, this was the age of the father. In this case, it's the age of the mother. So we get these paternal old father effects as well as old mother effects across taxa. Now here's some data from a long-term study. Again, this is working with this European shag species. Francis Daunt has a long-term study of these species, and we're able to look at offspring telomere loss during the fledging period in relation to parental age here. So I'm bringing it back to telomeres. Here's the mean age of the parents, 2 to 6 years, 7 to 11 years, or over 12 years. This is a wild population, can't separate maternal and paternal effects very well because the age of the parents tends to be correlated. But what you can see is that the telomere length of the offspring in the older parents was less at fledging than the telomere length in the younger parents at the time of fledging. And that was not related to growth rate. There was no difference in growth rate, but interestingly, there was no difference at hatching. So we think that this was some kind of postnatal effect, perhaps related to some aspect of parental behavior, but we haven't investigated that effect as yet any further. Now, that kind of study that I showed you there, and in many studies of these effects have been cross-sectional. That's to say different age classes have been compared. So what's important is to have a look at within individual studies to separate change in age, effects that come from changes in parental age, from differential survival of particular phenotypes into old age. We've been able to do that to a degree with our zebra finches. So here's this is again looking at telomere length, offspring telomere length here. Here's some work with a postdoc, Jose Noguera, who worked with us for a number of years. He was interested in the effect of old fathers. What he did in this experiment, he was interested in the prenatal effect. So he mated the same female zebra finches in quick succession with young and old males, and he randomized the order. So he obtained embryos from the same females who had been mated with old fathers and with young fathers. And here, so old fathers here and young fathers, same females, female age changed very little because it was done very quickly. And here you can see the embryo telomere length just after a few days of incubation in constant conditions in an incubator. And you can see that the embryos from the old fathers had significantly shorter telomeres. So this is pointing here to some kind of gametic effect that's taking place. Now here's something we did with the zebrafinch mothers. This here, again, offspring telomere length. These are the same mothers when they were 6 months old and when they were 3.5 years old. So this here is a within individual effect. We held the age of the male constant. So the males were young or relatively young. Obviously they weren't the same males, but these females have been mated with relatively young males when the females were 6 months old, and then again when they were 6.5 years old. And this is the telomere length in their offspring. And you can see that when the females were old, there was a very substantial reduction in offspring telomere length. We saw this, I said, I've been telling you about this experiment already, where we had the stressful and the environment and the benign environment. Found the same effect in both. I said that the age effect overwhelmed that sex difference in offspring that we saw when they were young. There was no sex difference in the offspring. Both sons and daughters showed this marked reduction in telomere length. Now, this is the offspring telomere length at fledging. So this could be a pre or a postnatal effect here. But remember, these are breeding in very benign conditions. They have ad-lib food when they're breeding. So shortage of food or aspects of parental behavior may not be important, but animals behave, they do many things. They brood, they incubate and so on. So it could be other effects going on there. One thing that we also found in our experiment when we had individuals in the challenging environment and in the control environment, we looked at levels of DNA damage in the red blood cells. This is done here with another postdoc, Antoine Stier. We were looking at this still quite early in life, when the individuals were only 5 months of age and when they were 1 year old. And you can see that, at least in the red blood cells, even in the control individuals, we found an increase in the level of DNA damage indices. And it was more marked in the individuals living in the challenging environment. So it's possible that that effect became even more marked in old age, and it may have influenced gametic performance in later life. But we don't know that as yet. But one thing we did find was that in the control birds, interestingly, the birds with higher levels of DNA damage at a younger age, in this young life measurement here, they also died at a younger age. And we did not see that effect in the individuals that were living in the challenging environment. We didn't see that the levels of DNA damage were linked to an earlier death. And that may relate to, in some somewhat complex way, may relate to resilience, but there's obviously a lot still to explore there. One of the things, just to finish up, that we've been coming on to look at is mitochondrial performance. That's obviously very important, and I've been doing some pilot work on this with some colleagues in Glasgow, Neil Metcalf and Colin Selman, but also in particular with two other postdocs here, Neil Dawson and Amelie Crespel. This just shows you some of the results. We were just here looking at changes in mitochondrial performance in relation to female age. This is ROS release by the mitochondria in two states: when the mitochondria are producing ATP to generate energy, and when they're not producing ATP. And all I want you to see is that in both mitochondrial states, the ROS production is higher in the older individuals. And that might mean they're generating more free radicals. But there's a lot to look at here in mitochondrial function. So I'm now pretty much at the end of my talk. As Elizabeth said, I just heard very recently that I got funding for a new project, which will start in the autumn of 2021. And it's focused on some of these maternal age effects that I've been talking about, and also variable environmental conditions, offspring outcomes, and mechanisms, including mitochondrial telomere interaction. So I'm going to be looking for a postdoc to work particularly probably on the mitochondrial function aspect. So just to conclude, environmental conditions can have long-term consequences for aging and longevity. Elevation of stress hormones in early life can shorten lifespan, and it also shortens telomeres. Importantly, outcomes of stress exposure can vary with age and environmental circumstances. We detect pre- and postnatal effects of parental age on offspring telomeres, whether that's coming through resilience or non-resilience to stress exposure and stress exposure itself. We don't know. And we have some suggestion that age-related changes in germline DNA may be involved in these effects of parental age on offspring telomere length, and possibly also on offspring longevity. So that's me finished. So thanks very much again for listening, and thanks again, Elizabeth, for inviting me. So
Thank you very much. Thank you.
So I stopped sharing my screen, Elizabeth.
Yeah, that's fine. Great, perfect. So thank you, Pat. That was a really fascinating and enlightening talk, and a real journey through the different aspects of your research, both focusing on the effects of the early life stress, but also how maternal and paternal effects can also play a role, and particularly as you show how timing can be important. So whether it's timing of exposure to stress or timing of the consequences further down the line. And it was quite nice how you showed, if you look at early life, early adulthood, or middle life adulthood, or later life adulthood, sometimes the effects you see can be very different. So yeah, that's really interesting and fascinating. So the questions are starting to come in from the audience. So while we give them a little bit more time, I'll kick off with a question for myself. So I'm quite interested because obviously you said that this research area hasn't really been addressed from a more evolutionary ecology perspective quite so much. And I was curious about the possible adaptive potential of these responses to early life stress. So you talked about how it could make individuals more resilient if they're more responsive to a particular stressor. But I wonder how this could interact with the frequency in which the environment changes. So, for example, if there's a mismatch between the early life environment and the adult environment, how adaptive would it be for individuals to be highly responsive to a stress which they may not actually experience in adult life? And I wonder if you could talk a bit about that. If possible.
Yes. I mean, that's a very good question and a very interesting question. Because of course, especially for long-lived species, the environment is probably not going to be stable, and it may vary across years, it may vary seasonally, and so on. So one might think it doesn't make sense to permanently adjust your phenotype in response to early life conditions. So some of these things may not be strategic adjustment. It may actually be a constraint that there may be setting of threshold responses at critical periods in development that are changed by exposure to stress in early life that cannot be undone, even if they are maladaptive. I mean, we as behavioral ecologists tend not to think so much about constraints because we tend to think, okay, evolution will fix it if it's going to reap fitness benefits, but there'll be a complex interplay between the costs of certain physiological changes, the probability of encountering a variable environment, and the developmental mode and the windows that open and close during development, and the costs of having a phenotype that's plastic, which is also very interesting. So there's scope for lots more experimental and genetic work on that kind of thing, I feel. The trouble with working with long-lived species is it takes a long time to get these effects. And maybe, well, if you go well down the line, I showed you even in the zebra finch, the results you get in the first year about longevity. If we stopped then, we just said it doesn't affect it. In middle age, it affected it one way, and in old age, it looked different. Obviously, the individuals who are alive at these points are changing a bit also, and it's complicated. You can only die once, so it's quite challenging to design good experiments to look at that.
Definitely, yeah. I think, as you say, there's a complex interaction between all these different factors. And do you see any sex differences in the response of the parental generation to these early life stresses? As you showed, I think with the offspring there were sex differences, but I wonder whether at the parental stage you see any.
We didn't see sex differences in the acute response. One thing I didn't talk about is when we, in that experiment where I showed where we treated them in early life and then looked at their survival, we also bred these individuals in different combinations, early stress and non-early stress and so on. We didn't find a sex effect on reproductive performance, but we did find that if your partner was an early stressed individual, your survival was reduced. So there was a transmission in adulthood presumably through the behavior of the exposed individual that somehow influenced the partner's behavior and influenced mortality rate. And it appeared to be an additive effect. So I actually have a postdoc who's been working with me over the past couple of years, who's looking at these behavioral changes in these individuals when they're adults. And she will also be looking for sex effects, but those partner effects were not sex specific. They could be transmitted by an early stress male or an early stress female.
It's quite remarkable. Surprising to see that, actually. So we have some questions from the audience. I'll move on to them now. So the first question asks, you mentioned that telomere lengths vary between species, but I was wondering, do you see much variation in telomere length rate of telomere loss within species, both in wild populations of animals and between wild versus domesticated individuals of the same species?
If I just address the first part of that first, yes, you do see a lot of individual variation, both in initial telomere length, if you like, and also in the rate of loss. And there have been a number of studies showing that the rate of loss is predictive of longevity. That's particularly so in birds, where there's most data, because birds have this great advantage of the nucleated red cell and the fact that you can get relatively non-invasively a sample where you can get DNA, and also birds are conspicuous and easier to follow longevity from ringing studies and so on. So yes, there is evidence of that. It doesn't appear to be the case in all taxa. There are taxa where we see much more telomere restoration in somatic cells than we do appear to see in many, but again, not all of the birds' mammals. So I haven't talked about telomere elongation and restoration, and that's a whole separate story. And I haven't been looking at that myself in great detail, but others have. The domesticated versus captive species is an interesting one. Again, that's complicated. And things seem to may happen to telomere length when a species becomes domesticated. For example, a laboratory mouse has much longer telomere length than the wild mouse of the same species. We've looked at telomere length in captive compared with wild zebra finches relatively recently, and there is no difference there. Domestic hens have very long telomeres also. People are interested in dog telomeres now because of the dog breeds. So there are interesting things to look at about the process of domestication and what are we selecting for that might produce these long telomeres in some species, right? It's hard, not being studied a lot, but there's a nice project to be done there.
Definitely, that's great. Is there any indication in your experiments as to why the sun's telomere lengths were affected more by the maternal stress exposure?
No, not really. I mean, one thing it could be linked to is if there was different growth dynamics in the males and females. We tend to find that we know, for example, that the tempo of growth can influence telomere loss, depending on how it's occurring, of course. But no, I haven't got a good answer to why that might be so. Obviously, often in a number of studies, especially in mammals, males sometimes seem to be less resilient to stressors than females. But again, that I don't want to make any generalizations that I haven't got data to back up with. So in answer to that question, I'd just like to say at the moment, don't know.
Yeah, okay. Someone's interested in the part of your talk where you talked about the mitochondria. And the question asks, given the age and higher oxidant effects, have you tested for higher mitochondrial number in older birds or higher mitochondrial DNA heteroplasmy? And if that might explain less efficient ATP production, perhaps?
I think it's a really interesting question, and that question is going to be addressed in the project that we're just about to start. So,
brilliant.
I don't have the answer, but I hope to have.
Great. Watch this space. And I think we'll just end with one final question for today. So, what role do you think epigenetic programming plays in the observed maternal effects?
In the effect, I guess you mean, well, there are two effects we could look at there. The effect, the maternal stress effect. One thing that we didn't do, and it would have been interesting actually to have a look at the acute stress response in the offspring of those females, and to do some complex cross-fostering would be nice, to look for any evidence of any transfer. Of course, you would need, it could be still be direct because of the way the parents are behaving with the nestlings in the nest. You'd want to go on another generation or two to look at that. There may well be epigenetic effects there, but in a single generation also. But in relation to the maternal age effect, again, there's lots to look at.
Yeah, definitely. I think that's a whole other research area, I guess.
And a very interesting one.
Definitely. Great. Well, I think that's all we have time for today, but it's our great pleasure to thank you once again for a truly fascinating talk.
Thank you very much.
Thank you. And for everyone else watching, our next talk will take place in 3 weeks' time on Wednesday, the 19th of May, when we will be joined by Dr. Alexandro Gonzalez Foya from UNAM University in Mexico, who will present an exciting talk on brain evolution. So save the date, and we hope that you can join us then. And in the meantime, check out our updates on our Slack and our Twitter feeds and spread the word about our seminars. Thanks for taking part, and until next time, stay safe and see you again soon. Goodbye.